Good afternoon, and thank you all for joining. I'm Cynthia Benz, and I serve as Vice President of Public Policy at the Alliance for Aging Research and Executive Director of the Accelerate Cures and Treatments for Alzheimer's Disease, or ACT-AD Coalition, and I'll be the moderator for today's program. On behalf of ACT-AD, it's my pleasure to welcome you to our fourth in a series of webinars focused on issues that are critical to the future of Alzheimer's disease research and therapeutic development. Today, more than 5 million Americans live with Alzheimer's disease. Because we don't yet know how to prevent Alzheimer's, as the population ages, the number of people with the disease is expected to reach 13.8 million by the middle of the century. Nearly 500,000 seniors are expected to develop it in 2017, and the Alzheimer's Association placed the cost of caring for AD at $259 billion this year alone. These costs will rise to $1.1 trillion by 2050 in the absence of a new medical breakthrough. ACT-AD is a coalition of more than 50 national nonprofit organizations working with urgency to speed up the development of potential cures and more effective treatments for Alzheimer's disease. And we do this by urging the FDA officials and other policymakers to increase the attention and resources they devote to the challenges posed by AD. Until the formation of ACT-AD in 2005, there was no single point of advocacy in the regulatory space around the need for better treatments to combat Alzheimer's disease that combine the perspectives of respected advocates for research, women's health, aging services, and caregivers. ACT-AD is strengthened by the diversity of the organizations that make up the coalition, and we're bringing that strength to bear on critical issues before the federal government concerning the development, review, and approval of a new generation of Alzheimer's disease therapies. One action FDA took in 2006 in response to ACT-AD's advocacy was to begin working with the coalition on an annual meeting where representatives from the FDA could interact with leaders in the patient advocacy community, academia, and industry on issues that are current roadblocks and potential roadblocks to the successful review of new Alzheimer's disease products. ACT-ADs convene 9 of these high-impact meetings on topics ranging from measuring clinical meaningfulness in AD, improving the efficiency of phase 2 trials, pursuing combination therapy development for Alzheimer's disease, and assessing the scientific foundation for AD drug development. ACT-AD will hold its 10th meeting with the FDA on November 16th, looking at the earliest behavioral and neuropsychiatric changes that occur with AD. And we hope that many of you will join us for that meeting, and the details will be available at the end of this program. Alzheimer's disease is often recognized by individuals or their loved ones when they experience memory loss or things like challenges with conversations. Eventually, the disease leads to an inability to respond to the environment around us. People with the disease require increasing levels of care as the disease progresses because they find themselves unable to perform even the most routine tasks. As the disease advances and more care is required, family caregivers often suffer from a decline in their own health while attending the needs of a spouse, parent, or other loved one with AD. Alzheimer's is a progressive disease that involves parts of the brain that control thought, memory, and language. Its development and progression varies from person to person in different settings, and researchers are still learning why this occurs. While there are clues on how best to intervene in the disease course, a revised framework is necessary to assist with research. So the National Institute on Aging and the Alzheimer's Association have been working to define Alzheimer's disease as a pathophysiological construct, much like osteoporosis or diabetes. For these diseases, biomarkers alone define the presence of the disease in a living person regardless of their symptoms. Clinical symptoms then assist in staging the clinical severity of the disease. The process of developing a research framework to define Alzheimer's across a continuum has involved the Food and Drug Administration, and ACT-AD's primary interest is in how this evolving definition of Alzheimer's disease as a continuum for research will ultimately impact therapeutic development. The NIAAA Working Group shared an update on the proposed framework at the Alzheimer's Association International Conference in London, England last week. During the session, they were provided with feedback that will help them in continuing to refine the final product, which is expected sometime in December of this year. Before the final release, the working group will provide at least 2 additional presentations of the draft research framework, and they're receiving comments on the framework from the public, and you'll hear more about that a bit later. And during today's webinar, Perspectives on the Alzheimer's Disease Continuum, 2 stellar experts who are true thought leaders will share their views on the role of biomarkers and clinical symptoms across the AD continuum, differences between the research framework and the 2011 NIAAA diagnostic criteria for Alzheimer's disease, and the important role this framework will play in future Alzheimer's disease research. We're very fortunate that both of them took time out of their schedules to be a part of this webinar. Before I begin, I'd like to say how grateful we are to ACT-AD sponsors, Anavex, Avenir, Axovant, Biogen, Eisai, Eli Lilly, Genentech, Janssen, Lundbeck, Merck, Novartis, and Otsuka. Without your generosity, programs like the one today would not be possible, so thank you very much. I'd also like to thank my colleagues Sarah DeGiovane, Ryan Carney, Noel Lloyd, Brianna Bishop, and for their assistance with the webinar. And last, I just need to take care of a few housekeeping items. There are more than 100 registrants on today's webinar, so we've automatically muted the lines. You can submit a question at any point throughout the webinar using the chat function or by emailing me, and my email address is cbenz@agingresearch.org. And each presenter is going to speak for approximately 20 minutes. We're not going to be taking questions at the end of each presentation. We're actually going to be holding questions for the Q&A period at the end of the webinar. We're going to answer as many questions as we can before we close the program at 4 p.m. But if your question's not addressed, please email me and I'll work to try to get your questions answered by email. We're recording today's program, and we'll try to make the recording available no later than 1 week after the webinar is concluded. So I have the pleasure of introducing our first speaker, and we're actually going to start with you, Paul, if that's okay. Dr. Paul Azen is founding director at the Alzheimer's Therapeutic Research Institute at the University of Southern California's Keck School of Medicine. Dr. Azen's been a leading figure in Alzheimer's disease research for more than 2 decades, having developed novel methodologies as well as designed and directed many large therapeutic trials. He received his BA in biochemistry and molecular biology from Harvard and his medical degree from Columbia. He completed his residency at Case Western Reserve University in Cleveland and Mount Sinai Medical Center in New York, and then fellowship training in rheumatology at New York University. After serving as chief medical resident in Mount Sinai, he began a solo practice in internal medicine and rheumatology in New York. Dr. Azen joined the faculty of Mount Sinai in 1994 and was recruited to Georgetown University in 1999 as professor of neurology and medicine. That year, he founded the Memory Disorders Program, a clinical and research program for Alzheimer's disease and related disorders. He continued basic research studies on therapeutic targets and biomarkers and designed and directed multi-center therapeutic trials. He became vice chair of the Department of Neurology at Georgetown in 2004. From 2007 through 2015, he was professor in the Department of Neurosciences at the University of California, San Diego, and director of the Alzheimer's Disease Cooperative Study. Dr. Azen sits on ACT-AD Science Advisory Board, and he's been instrumental in helping the coalition identify issues for our annual meetings and webinars that are of interest to the various audiences we interact with, from patient advocates to regulators. He really is the reason why we chose this topic for the webinar today, and it turned out to be a hit with our followers. So you have my personal thanks for that, Paul. And so with that, I'm going to pass the presenter control, so take it away.
Thanks very much for that kind introduction, Cynthia. It's a pleasure to be able to speak to this audience today about our current view of the spectrum of Alzheimer's disease and thinking in terms of a continuum rather than stages. There are a few basic points that I'd like to make during this session. I'd like to present this view of AD that differs from our traditional look, which considered discrete clinical stages, first mild cognitive impairment and then dementia. I'm going to try to demonstrate the reasons that we think progression of Alzheimer's disease should be considered a continuous process that is best measured by scales that show this continuous progress, because considering movement from one clinical stage to another throws out most of the information that describes the gradual worsening of the disease. I'm going to talk in particular about measuring cognition as an indicator of the decline of Alzheimer's disease, and show that cognitive decline occurs quite early and can be used to mark progression throughout the entire spectrum of Alzheimer's disease. And I'm going to present a simplified view of the disease as a process that begins with the accumulation of amyloid in brain, but is marked by cognitive change across its entire perspective. And I'm going to talk about the implications of this view on drug development and how this increases the likelihood of our achieving significant advances in treatment. This slide shows our traditional view of the clinical progression of Alzheimer's disease. So we marked the onset of the disease, shown in this figure as a vertical dotted line. We marked the onset of disease as the onset of the syndrome of dementia, that is, cognitive impairment significant enough to interfere with daily function. We did talk about stages of dementia, mild, moderate, and severe, but the entire disease was considered to be a dementia. And so symptoms and biomarker evidence of abnormalities prior to the onset of dementia were relegated to a relatively nonspecific categorization as mild cognitive impairment, meaning cognitive impairment that is not serious enough to impair a daily function. So we talked about a 7 to 10 year progression of disease from the onset of mild dementia through gradual worsening to severe dementia and loss of independence and eventual death. But the period before dementia, the MCI period, was always a heterogeneous clinical construct, and that was quite problematic in that it was not accepted by regulators as a disease. So mild cognitive impairment was considered to be a syndrome that could represent any one of several diseases: Alzheimer's disease, vascular disease, Lewy body disease, for example. And therefore, drug development in the pre-dementia stage was difficult, and the only acceptable endpoint was time to the onset of Alzheimer's dementia. And that is a difficult endpoint in that it throws out continuous measures of cognitive worsening and therefore presents a barrier to demonstrating drug effects. We now think of AD as a continuum. While the clinical stages that I've mentioned, mild cognitive impairment and dementia, remain very useful in clinical practice, we have essentially discarded those constructs, MCI and dementia, in the drug development world in Alzheimer's disease and in the clinical research efforts in general. We now use biomarker-confirmed stages of disease, specifically preclinical Alzheimer's, prodromal Alzheimer's, and Alzheimer's dementia, but in each case defined by specific biomarker stages. We use this terminology for selecting individuals for trials, but we do not use the stages as outcomes in trials, as we were forced to do when we had studies in mild cognitive impairment. Now we are able to talk about the entire continuum, starting from an asymptomatic stage, preclinical AD, through the MCI stage that we now call prodromal AD, anchored by biomarkers of amyloid accumulation in brain, all the way through to Alzheimer's dementia. And regulators have embraced this continuum approach, and that allows us to use much more sensitive, informative measures of disease progression and drug effects that, again, increase our likelihood of successful drug development. I'll be focusing on cognition, but biomarkers play a critical part of the AD continuum in clinical research. We place people on the AD continuum, that is, identify people who are somewhere on the AD spectrum by using one type of biomarker, brain amyloidosis. This is actually the onset of the AD continuum that can be defined or measured in two different ways. We use amyloid PET scanning to visualize the accumulation of amyloid in the brain. We can also measure amyloid peptide in the cerebrospinal fluid obtained by lumbar puncture, that is spinal tap. And using either of these two techniques, PET scanning or spinal tap, we can accurately place individuals on the AD continuum. So by these biomarkers, either PET or CSF, we can accurately place an individual on the AD continuum. But measures of amyloid don't indicate where on the continuum, how much progression has occurred in an individual. For that, we can use other biomarkers. And the most exciting new biomarker that helps establish the state of progression or the position on the continuum is tau PET scanning. So another type of molecular imaging, in addition to amyloid PET, tau PET scanning actually indicates severity. It places individuals at a specific spot on the continuum. So we identify those that are on the continuum with amyloid measures and then can place individuals in a specific position using tau PET or other biomarkers. For example, FDG PET, which indicates metabolic activity in the brain, or volumes of brain structures such as the hippocampus that can be measured using MRI, or some biochemical measures in the cerebrospinal fluid obtained by spinal tap. But I want to suggest that cognitive measures themselves may serve this purpose. They can place people at a specific position on the AD continuum, and measuring cognition may even be considered a biomarker itself, and in some ways is the ideal biomarker for placing individuals at a specific position along the AD continuum. Well, why do I say that? Well, first, of course, the cardinal symptoms of Alzheimer's disease are symptoms of cognitive decline. So measuring cognition stands as the most face-valid measure of degree of impairment from Alzheimer's disease. And when we're doing drug studies, we want to improve cognition. So it is an excellent biomarker of drug response and is generally part of the outcome measures of our clinical trials. We have tried to use biomarkers as outcome measures, but sometimes biomarkers can be confusing. They sometimes move in unexpected directions in response to treatment. An example is an unexpected increase in atrophy that can occur as amyloid is removed from brain and inflammation recedes, shrinking volumes. We always know that we need to improve cognition. It's got the clearest directionality as an outcome measure, and it is useful across the entire spectrum, from the onset of the continuum that is marked by positivity on amyloid measures, amyloid PET or CSF measures, and it remains dynamic, that is, it changes as the disease progresses all the way through end-stage disease. And this is particularly valuable for drug development because it links studies at different points in the disease with similar outcome measures measuring cognitive performance, and it supports the combination of different trials at different stages of disease, from the asymptomatic stage through the mild cognitive impairment stage all the way through to dementia, and supports registration packets for consideration by the FDA that include trials at very different stages of disease. Now, one of the shortcomings of using cognition as a measure, well, one, as mentioned on the previous slide, is that it's very variable, and so noise confounds the use of cognition, and we're constantly working to improve the measurement characteristics of cognitive measures. That's one drawback, but a second is that a cognitive measurement by a memory test, say, does not necessarily indicate clinically meaningful drug effects, and that's why traditionally the FDA and the EMA have required co-primary measures in drug studies, a measure of cognition and a measure of clinically meaningful change like function or a clinician's impression of change, co-primary measures for clinical trials. But as we've been moving our clinical trials to the earlier stages, the asymptomatic stages, the earliest part of the continuum, we have a problem because if an individual is asymptomatic, if they have no functional impairment, how do we establish clinically meaningful benefit from drug intervention? And we've been working to establish the predictive value of drug effects on cognition as far as later functional decline is concerned. And there's been significant progress here that has opened the regulators to the idea that a single cognitive measure may allow for at least accelerated approval of a drug. And that's a major advance. Now, Cliff Jack's going to be talking in a few minutes, and Cliff really moved the field along substantially with his description of Alzheimer's disease in terms of a sequence of changes of biomarkers and cognitive and functional measures. The Jack diagram that I'm showing here is one of the most widely used tools in AD clinical research. But I want to suggest a slight modification to this view of the disease. So as Cliff has shown here, we say that changes in measures of amyloid occur first. They're shown in the red line. And changes in function occur last. They're shown in the green line. And we all agree on that part of this curve. But the curve suggests that cognitive change, here indicated as memory and shown in the, I guess it's purple curve that is towards the right just before the functional change. This diagram suggests that cognitive change or memory change is late in a sequence of biomarker and clinical measures as AD progresses along its continuum. And I think that we may want to refine that a bit. We now know, and we've known for some time, that cognitive measures actually can mark very early decline in the AD spectrum. And this is actually an early demonstration of this a decade ago from a long-term observational study conducted in France called the Pathophysiologic study that showed that individuals who eventually develop dementia separate from individuals that are free of dementia 14 years before the onset of dementia just by measuring cognition, even with a gross tool like the MMSE, the Mini Mental State Examination. So this shows that memory decline or cognitive decline is not a late measure, but an early measure of the AD spectrum. And we've been trying to use a data-driven approach, actually measuring and ordering the sequence of changes using the rich data set that comes from the Adney Study. And while this is challenging, I've shown here our latest version of a data-driven ordering of changes. And without trying to explain every line here, I will say that this figure agrees that amyloid changes come first and functional changes come last. But by and large, biomarkers of tau accumulation or tangle accumulation and atrophy of brain structures and loss of metabolic brain function and cognition are all roughly superimposed between the amyloid accumulation and the functional decline. That, in fact, it's not an ordered sequence of changes, but rather very much superimposed curves representing biomarker and cognitive changes as the AD continuum progresses. And so here I've drawn a much simplified version of Cliff's diagram, where I just show that amyloid accumulates first, as shown in red, function declines last, as shown in green, and in between we can simplify the curves I just showed by saying that there is a superimposed gradual decline in biomarkers such as FDG PET or tau PET or volumetric MR scanning and cognitive change, that all of these move roughly together in a superimposed way once the amyloid has deposited in the brain to start the process all the way through the functional decline that characterizes progressive dementia. And we have cognitive and clinical tools that can place people at specific points on the AD continuum that just focus on cognition and symptoms. And I've summarized them here. I'm not going to go through all of them, but I will say that there are brief, useful tests of early symptoms and cognitive change that could be used in a clinical setting, more detailed measures that are used by neuropsychologists to place people at points on the continuum. And in drug development work and clinical trials, we now have good tools that assess the early cognitive change sensitively. And an example is the PAC, which is a cognitive composite useful at the asymptomatic stage of disease. We have useful outcome measures in the prodromal or MCI stage and in the dementia stage that allow us to use cognitive decline and measurement of symptoms to accurately measure drug effects without the gross tool of using time to a next clinical stage, which throws out so much information. So this formulation gives us much more sensitive tools for drug development. But one of the questions that has arisen as we try to use this continuum construct to move our trials of potential disease-modifying drugs, and especially anti-amyloid drugs, to a very early stage of disease, a major question has arisen: how do we know for sure that once someone has amyloid, they are going to be on this continuum and will progress to the symptomatic stage of disease? This is critically important for drug development because, as I said, we're using this construct of continuum, this placing of people on the AD continuum just using amyloid measures. We'd better be confident that an amyloid abnormality, whether by PET or by cerebrospinal fluid, does in fact specifically indicate that someone is on the continuum and is going to progress unless there is some other intervening illness and an individual does not survive long enough. An individual who's on the AD continuum indicated by an amyloid abnormality is going to progress to symptomatic Alzheimer's disease. We've just completed an examination of data from the Adney Study, from the Alzheimer's Disease Neuroimaging Initiative, that specifically looks at this issue. It takes people who start out entirely clinically normal and divides those normal people into two categories: people who have evidence of amyloid accumulation by PET scan or by examination of CSF, or people who do not have those abnormalities. They are normal in terms of amyloid negative. And it examines the idea that those who have elevated amyloid, shown here in red, compared to those who do not have elevated amyloid, have a different trajectory. So all these individuals from the Adney normal cohort start off at the 0 time point on the left of these graphs as being clinically entirely normal. They were recruited as normal controls into Adney. But some of them had biomarker evidence of amyloid despite the fact that they had absolutely no symptoms, no cognitive concerns. They were clinically normal. We've now followed individuals for years, sometimes up to 10 years and longer, so we can examine the impact of amyloid in clinically normal people, the impact on their long-term cognitive health. And we see here that using the mini mental state exam, the MMSE, on the left, on the right, as was used in that Packwood study, does show that people who don't have amyloid decline very little, even over 10 years, whereas the decline is much more substantial in people who have elevated amyloid. And we are able to more accurately separate the groups based on cognition using better cognitive tools. Here, the PAC that I mentioned that we call the Preclinical Alzheimer's Cognitive Composite. The PAC actually shows separation within 3 to 4 years of people who start out clinically normal, those with amyloid separated from those who don't have amyloid. Using the MMSE, it takes a bit longer, but both cognitive tests show the distinction based on the presence of amyloid alone. Now, this is cognitive decline. We can also look now over a 10-year period at clinical decline, at decline marked by manifestations of symptomatic Alzheimer's disease. So here we are using early mild cognitive impairment defined in terms of episodic memory impairment and in terms of clinical staging with the clinical dementia rating, and showing that at the end of 10 years of follow-up, the clinically normal people who did not have elevated amyloid at the outset remain clinically normal. That is, 90% are still clinically normal at the end of 10 years, whereas those that had elevated amyloid at the outset, by the end of 10 years, over 80% meet all of the criteria for early MCI related to Alzheimer's disease. So the great majority of people after 10 years have symptomatic Alzheimer's disease. Preclinical AD, clinically normal individuals who have elevated amyloid will progress to symptomatic Alzheimer's disease. We're still collecting more information. We only have a small number of people who have been followed in Adney for 10 years, so we continue to collect information, but we think this picture is very clear that the continuum begins with the abnormality of amyloid, and people on the continuum will progress to symptomatic Alzheimer's disease. This is very important for anti-amyloid therapies because we think the earlier we intervene, the better, and we believe we can accurately identify people on the continuum on the basis of amyloid biomarkers. That's the preclinical AD construct. We also believe that we can now remove amyloid from brain using certain types of antibodies like aducanumab, and this means that we have the potential for removing amyloid at a specific but asymptomatic stage to prevent or greatly slow the development of symptomatic Alzheimer's disease. And I've just shown here a figure from a paper published last year using this aducanumab anti-amyloid antibody that shows on amyloid PET scans you can take someone with a brain full of amyloid shown on the left-hand column as red, and at the end of 1 year at the highest dose shown in the bottom figure on the right-hand column, you can remove essentially all of the amyloid. If we take this treatment that removes amyloid and intervene with it at an asymptomatic stage when the brain is functioning normally, we think we have an excellent opportunity to dramatically alter the continuum of AD. And so that's shown here on this figure. Again, the simple curves that I showed you that we need to intervene here on the left, shown in that column on the left, orange column, where amyloid is elevated, and that's where we need to attack the amyloid and remove it before there is substantial cognitive decline or changes in other biomarkers, and that's going to be our path to preventing the decline in clinical function that characterizes symptomatic Alzheimer's disease. So in summary then, we believe that the continuum gives us the best framework for drug development, allowing us to use continuous measures that are much more powerful in assessing drug effects. We believe that even the asymptomatic stage of AD, where people are clinically entirely normal but have measures indicating amyloid accumulation in brain, is a specific part of the AD spectrum that is appropriate for disease modification trials, even at anti-amyloid trials that carry some risk. Because we believe that the evidence is strong, that without treatment these individuals are going to progress to symptomatic Alzheimer's disease, we think it is entirely appropriate and even essential that we bring our best anti-amyloid interventions to preclinical AD. We believe that the continuum allows greater flexibility in putting together packages for submission for FDA and EMA approval, linking drug effects at all stages of disease by their impact on cognitive performance. And in my final conclusion, I think that this continuum approach greatly increases the likelihood of successful development of disease-modifying strategies for AD. And thanks very much for your attention.
Thank you so much, Paul, for that really comprehensive presentation. We really appreciate it, and we have had a couple of questions come in, so we're looking forward to getting responses to those at the end of the program. And now I have the pleasure of introducing Dr. Cliff Jack. Dr. Jack is Professor of Radiology at Mayo Clinic in Rochester. His lab is engaged in brain imaging research and cognitive aging, Alzheimer's disease, and related disorders. Dr. Jack's research team employs MRI and PET to study the biology of brain aging and the causes of cognitive impairment. They develop imaging processing algorithms for quantitatively measuring the information obtained from brain imaging. Dr. Jack's team employs a variety of MRI-based brain imaging modalities, including structural MRI, spectroscopy, functional connectivity, and brain water diffusion. They also employ FDG PET and amyloid PET. The lab's clinical imaging research is tightly integrated into the Mayo Clinic's Alzheimer's disease patient registry and Mayo's Alzheimer's Disease Research Center, which conducts NIH-funded longitudinal clinical and epidemiological research projects investigating normal aging, Alzheimer's disease, and other dementias. Dr. Jack's been intimately involved in the process to develop the revised framework, and he presented the revised research framework on behalf of the NIAAA working group at AAIC. We're very fortunate to have him join us to talk about the process and how the community can continue to engage. So Dr. Jack, please take it away. And you should be able to advance your slides by just clicking the arrow at the top of the page.
Okay. Thanks very much. I'm looking for the arrow.
Okay.
I'm looking,
it should be there now.
Finding.
Okay. Well, I can go ahead and advance the slides. Just say next slide.
Wherever the arrow is, I don't see it. So, sorry.
No
okay.
problem.
Thanks very much for inviting me. And actually, I think thanks for pairing Paul and I together, because I think our presentations are going to be very complementary. So, and as I go through this, I'll try to link what I'm saying back to some of the points that Paul made. So one of the points Paul made in his presentation was that the historic definition of Alzheimer's disease began when symptoms began. And you'll notice in the title of this presentation, an NIAAA research framework, towards a biologic definition of Alzheimer's disease. The National Institute on Aging and Alzheimer's Association Committee, or work group, to update and revise the criteria are actually proposing that the definition of Alzheimer's disease not be syndromal, but rather be biologically based. So could you please advance? The members of this work group are listed here. I won't read off all the names, but the membership in this work group was selected by the leadership of the National Institute on Aging and Alzheimer's Association, and the intention was to give this work group a broad range of technical expertise and background and geographic representation. So next slide. I guess I can see the arrows. Yeah, sorry, I can do it. Okay, so the objectives of the work group are outlined in this slide here, and the italicized words are key. So update a scheme for defining, staging the disease across its entire spectrum with which the research community, et cetera. So our objectives were to define, to stage, and this is intended only for the research community. So this is not intended for daily clinical use. It's intended only for research use. Okay. So everyone, I'm sure, is familiar with the 2011 National Institute on Aging Alzheimer's Association, or NIAAA Guidelines. And one could ask, why bother updating these? A lot of people seem to like them. And the reasons for updating the guidelines are severalfold. First, increasing acceptance that certain biomarkers are valid proxies for the underlying pathologic changes of Alzheimer's disease. Second, increasing recognition of the frequent mismatches between a clinical diagnosis of Alzheimer's disease and actual evidence of Alzheimer's pathologic change, particularly as a product of many biomarker studies that have been published since the original NIAAA guidelines were published in 2011. Next, recognition that Alzheimer's disease is a continuum. This directly ties in with what Paul was advocating, and that is regarding the disease as a continuum. An unfortunate product, I think it was certainly not intentional, but an unfortunate side effect, if you will, of publishing 3 separate documents in the 2011 NIAAA guidelines was that this was interpreted by some people to indicate that normal, MCI, and dementia were 3 separate entities. That wasn't the intention, but that it created that perception. And that is a perception that the current working committee strongly feels needs to be erased. Alzheimer's disease is a continuum, and it needs to be portrayed that way, as Paul has said. Another reason is the implementation of biomarkers needs to be harmonized. Conceptually, it needs to be identical across the entire continuum of the disease, from preclinical through symptomatic disease. That was not the case in the 2011 guidelines. So in the preclinical guidelines work group that was headed by Risa Sperling, amyloid biomarkers were placed at the top of the hierarchy. Being amyloid positive alone puts you into the Alzheimer's pathway, whereas for the symptomatic phases, the MCI and dementia phases of the disease, this was not the case. All biomarkers were treated equally. For example, an atrophic hippocampus was regarded to be equally specific for Alzheimer's disease as an abnormal amyloid biomarker study, and we of course know that that's incorrect. So that has had to be corrected. And finally, as Paul indicated, the most exciting development in the biomarker area in recent years is the introduction of tau PET, and we certainly felt that a modern research framework ought to have tau PET fully integrated into it. So what were some of the principles that the work group used to guide its deliberations? And I'll just repeat this again because it bears repeating, and that is this was designed to be a research framework only. It was not designed and was not intended for general clinical care. And we recognized that there were 2 general use cases for research, observational and interventional research. And I want to stop for a second and emphasize this. So some of the material that I'm going to present over the next couple of minutes might seem a little confusing with respect to what Paul presented, but Paul's focus was, rightly so, on interventional research. So his emphasis was how do you operate, how do you run clinical trials. But in a broader sense, clinical research encompasses both clinical trials but also observational research. And in observational research, in fact, one of the key aspects of the scientific questions that are asked involve comparing and contrasting people who have Alzheimer's disease with people who don't have Alzheimer's disease. And so all members of any research cohort are typically, or I won't say must, but are typically included in observational research. And so biomarker characterization and clinical characterization of the full spectrum has to include and identify who is in and who's not in the Alzheimer's pathway. So our work group took a stepwise approach to constructing this framework. We asked the question, debated it, answered it, and then moved on to the next logical question in sequence. So the first question is, what is the definition of Alzheimer's disease? And as I indicated, our work group indicated that the term Alzheimer's disease should refer to pathologic change and not to a syndrome or syndromes. Alzheimer's disease, therefore, is identified either at postmortem by pathologic changes, as it has always been, and in life by biomarkers, not by symptoms. Symptoms certainly are a part of the disease continuum, but they're not its definition. This obviously constitutes a major shift in thinking. So in 1984, the McCann criteria, Alzheimer's disease was envisioned as a clinical pathologic construct, the idea being that if one had the typical multidomain amnestic syndrome, you had plaques and tangles, and if you did not have a multidomain amnestic progressive syndrome, you did not have plaques and tangles. So that is obviously not right. It seemed logical at the time, but it's been proven in many cases not to be true. From 2007 through 2014, the IWG International Work Group published a series of 3 different position papers redefining Alzheimer's disease, and the NIAAA Guidelines were published in 2011. And these guidelines generally took the approach that Alzheimer's disease was defined as a clinical biomarker construct, meaning that biomarkers were used to increase or decrease the probability that Alzheimer's disease was the underlying etiology explaining the observed symptoms. And now in 2018, I say 2018 because that's when the document will be published, our position is that Alzheimer's disease should be regarded as a biological construct across the entire disease continuum, not a syndromal construct. So the next question then becomes, if Alzheimer's disease is defined for research purposes only, again, by biomarkers in living people, then what biomarker profiles should define Alzheimer's disease? And our position was that only biomarkers that are specific for hallmark AD proteinopathies, that is beta amyloid and pathologic tau, could be considered as potential biomarkers to define the presence of the disease. And there were some additional specifications. So whatever the biomarker definition was, it had to function equally well for disease onset that occurred in early life through late life. It had to function equally well for an individual from the pre-symptomatic through the symptomatic phases of this continuum. And it also had to function equally well for both typical and atypical clinical presentations. So this is what we came up with, definition of Alzheimer's disease. This is a somewhat nuanced definition, but we believe it is justified by the relationship between what's known about pathology and the biomarkers of those pathologic changes. So Alzheimer's pathologic change was the designation if there was biomarker evidence of A-beta amyloid alone. That is, this would be applied either when a tau biomarker was normal or when it was not available. The term Alzheimer's disease would be reserved for instances where there was biomarker evidence of both amyloid and pathologic tau. Why is that? The reason is that for a pathologic diagnosis or an autopsy diagnosis of Alzheimer's disease, both beta amyloid and evidence of pathologic tau beyond a certain Braak stage have to be present in order to meet pathologic criteria for Alzheimer's disease. So these were not regarded as separate entities, but rather earlier and later phases of what we term the Alzheimer's pathologic continuum. Again, this ties directly into Paul's conceptual picture of this, that the disease is a continuum, not compartments. And the biomarker definitions are independent from symptoms. So regardless of whether symptoms are present, absent, or if they're present, what the nature of the symptoms are, these biomarker definitions operate independently. Now, our second objective, first objective was defining. The second objective was staging the disease. Again, we had some guiding principles, and that is that the measures used to define the disease certainly had to be specific for the AD pathologic changes, but measures used to stage the disease need not be. So biomarkers then were used for both purposes. A-beta biomarkers determined whether or not an individual was in the Alzheimer's pathologic continuum, yes or no. If A-beta is present, then the person is in the Alzheimer's pathologic continuum. Tau biomarkers are used to make the designation Alzheimer's disease. If someone is in the Alzheimer's pathologic continuum by virtue of having an abnormal A-beta biomarker and they have an abnormal tau biomarker, then that's labeled Alzheimer's disease. Neurodegenerative and neuronal injury biomarkers, as Paul indicated, are not specific for Alzheimer's disease, and therefore these are used to stage the disease only. Symptoms, likewise, symptom severity, are not specific for Alzheimer's disease, and therefore they're used only to stage disease severity, clinical or cognitive severity. And biomarker and cognitive staging are done independently from one another. Some features now of operationalizing of biomarkers for definition and staging. There are a lot of different biomarkers in use in the research settings, at least, and we wanted to come up with a common framework so that investigators who use predominantly imaging or investigators who use predominantly CSF or investigators who preferred a mix of imaging and CSF could communicate in a common language or a common framework. And this is done by simply placing biomarkers into groups based on the pathologic process that each measures. And our work group followed the recommendations that had been published in a white paper last year, so-called ATN, and it's a very simple idea, and it's simply classifying biomarkers on the basis of what each is purported to measure. This is what the classification system looks like. So you can see biomarkers are broken down or divided into those of amyloid plaques or associated physiologic state, so-called A or amyloid in red. In yellow, you see biomarkers of aggregated tau or the associated physiologic state, so-called T biomarkers, and then biomarkers of neuronal injury and neurodegeneration, so-called N biomarkers. That's where the ATN comes from. And you can see that there's both a CSF and at least one imaging measure in each one of these categories. And so people can be classified fully either by CSF or by imaging or by mixtures of the two. Now, each of these biomarker groups can be, it doesn't have to be, but it can be dichotomized. And if you happen to do that, so plus minus, positive, negative, you wind up getting 8 so-called profiles, and you can see these 8 different plus minus profiles listed here under the column ATN profiles. And these 8 profiles in turn can be broken down or can be subgrouped into 3 biomarker categories. At the top you see A minus, T minus, N minus is normal AD biomarkers. The bottom 3 profiles in the dark gray are non-AD pathologic change. So that is, these are individuals who have a negative or normal amyloid study, but either the tau or the neurodegeneration or both biomarker categories register an abnormality. So there's something going on in the brain of these people, but it's not Alzheimer's disease. And then in the middle, 4 boxes, you see in light gray the profiles that we would label Alzheimer's pathologic continuum, and these are broken down again into those that would be labeled Alzheimer's pathologic change versus Alzheimer's disease. So I'm just going to show a couple examples here for people who aren't familiar with biomarkers, just to make these concepts maybe a little bit more concrete. So this is an example of tau, PET, amyloid PET, and MR images from a 33-year-old cognitively unimpaired individual. And these are three different axial slices through the brain, clustered: the lower slice on the left, middle slice in the middle, and the upper slice or higher slice on the right-hand side of the slide. And you can see, as you would expect for a 33-year-old cognitively unimpaired individual, that there's no uptake of the tau ligand in the gray matter. There's no uptake of the amyloid PET ligand in the gray matter, and the MR shows no atrophy. So this would be an A-, T-, N- profile. Again, what you would expect in a 33-year-old. This next case is the opposite, if you will. So this is a 75-year-old with a typical amnestic multi-domain dementia syndrome. You can see 3 tau scans across the top. You can see uptake of the tau PET ligand in yellow and red colors in the cortex, exhibiting a particular fairly typical topographic pattern of uptake. On the bottom left, you see the amyloid PET scan. Again, the uptake is indicated in red, so uptake is present diffusely throughout the brain with no particular topographic pattern. This is characteristic of amyloid. And then on MR, the bottom right, you see atrophy in the temporal lobes and elsewhere. So this would be an A+, T+, N+ biomarker profile as depicted by imaging. And lastly, in these examples, you again see 3 different slice locations, low, middle, and high. This is a cognitively unimpaired 67-year-old. The tau PET scan is negative, so on all 3 slices, there's no evidence of uptake of the tau PET ligand in the gray matter, but the amyloid PET scan is grossly abnormal. So you see diffuse uptake of the amyloid PET ligand throughout the gray matter, again, without any particular pattern, and the MR demonstrates not much atrophy. So this would be an A+, T-, N- profile. So the purpose of going through these slides is to give people a feel for what we mean when we say A+, T+, etc., and to indicate that, so these are visually obvious examples, but to indicate that there, even let's say among cognitively normal people, you will see a variety of these different biomarker profiles if you sample the population. And this case that we see here, cognitively unimpaired, 67-year-old, he was amyloid positive but tau negative and MR negative, is exactly the kind of individual that Paul was describing, who would be an ideal candidate for enrollment in a secondary prevention trial. So this is someone who clearly has amyloid in the head. They've not yet experienced significant cognitive decline, and the idea would be to arrest or reverse amyloid deposition. So let me move on now to cognitive staging, and it says in the slide 2 schemes, but in reality there are 3 schemes, and one is simply to use continuous measures of cognition, as Paul indicated, something like the MMSE or a cognitive composite, to treat the cognitive scale as a continuum. That's one option which we outline in the working document here. But our group has received a lot of feedback from investigators in the field, indicating a preference for 2 additional kinds of staging schemes. And one is called the syndromal categorical cognitive staging. That employs 3 traditional cognitive categories, so cognitively unimpaired, MCI, and dementia. These are largely the same, defined in the same way as the 2011 guidelines, so I won't go through these at all beyond this in any detail. Again, this is done independently from biomarkers, and it includes all members of any research cohort. So if one randomly samples the population, you will have in that sample people who will fall into each one of these three categories and will fall into all 8 of the biomarker categories. And the second, and so the syndromal categorical cognitive staging would be a logical approach in observational or natural history kinds of studies. The second staging scheme, this largely came, I would say, from people who were involved in clinical trials. It's so-called numeric neurocognitive staging. And here stages are broken into 1 through 6. I won't go through what the stages are, but you can imagine that things get worse as you go from 1 through 6. Actually, 1 through 6 are very, very reminiscent of the global deterioration scale of Barrier-Reisberg, published back in the 1980s, which was, of course, before biomarkers. The idea here was that we wanted to avoid traditional syndromal labels, cognitively unimpaired, MCI, and dementia, and simply give these stages numbers. This, importantly, is applicable only to those in the Alzheimer's pathologic continuum. So this is not applicable to all members of a randomly selected group from the population. It's only applicable to those who fall into the Alzheimer's pathologic continuum, which is the group that you would be interested in for a typical modern clinical trial. Now, we go back to the syndromal categorical cognitive staging scheme, and I just want to illustrate a couple points here in this table. And the point that you can see that this table represents the marriage of syndromal categorical cognitive staging with biomarker profiles. And the first thing that this table illustrates is the fact that these are 2 independent axes of information acquired about every individual research participant. So a research participant is placed into one of these three cognitive staging categories, and is also placed into a biomarker profile category. And those things are done independently. So the definition of dementia is independent of the biomarker profile and vice versa. The second point that this illustrates, I would say, this table illustrates, is our committee's attempt, or our work group's attempt, to honor the wishes or the suggestion of a number of investigators in the field who liked the descriptive phrases that you see in the body of the table. For example, many people in the field feel, rightly so, that terms like preclinical Alzheimer's disease is very useful. This is something that appeared in the 2011 preclinical guidelines document led by Risa Sperling. People like this term, they feel it's useful, and they would like to have it retained in the 2018 version of the NIAAA Guidelines. And I won't read through all the various descriptors in this table, but that's the nomenclature in this table is intended to bring these phrases forward from 2011. Another thing that this table illustrates, however, is that one can characterize research participants without using any of the phrases in the body of the table. Other investigators indicated to us that they don't like these phrases, and what they would prefer is simply to categorize research participants by the biomarker profile and the cognitive stage. For example, someone will be labeled A+, T+, N+, cognitively unimpaired, rather than preclinical Alzheimer's disease. Either approach is fine. Some people tend to feel very strongly about this, and our work group was agnostic. People are free to use whatever type of terminology they think is most appropriate, but both types of terminology are laid out in the document, the working document. The last slide I'll show is this: clinical research without biomarkers or with incomplete biomarker information. Cynthia, I think, mentioned that this was presented. This framework has been presented, I think, about 5 times now. The most recently last week at the AAIC meeting, and many people in the audience objected violently to the idea of defining Alzheimer's disease by biomarkers rather than using the traditional clinical definition of a multidomain amnestic syndrome that began in 1984. And I think people understandably would feel threatened if they were involved in research where it was just not feasible to do biomarkers, and they would feel that their research programs were threatened. But in reality, our intent is to indicate that this research framework is intended to be complementary to clinical research without biomarkers. Clinical research without biomarkers, we believe, is still valuable, and it's complementary to what's done with biomarkers. People who do not have access, or for whatever reason it's not feasible to use biomarkers in the research programs, can continue to use the traditional probabilistic terms, probable or possible AD, recognizing that there's about a 20% false positive rate in designating someone Alzheimer's disease clinically with respect to an autopsy gold standard, and there's up to a 40% false negative rate in designating someone who's clinically normal as not having Alzheimer's disease. But with these probabilistic caveats, there's nothing wrong with continuing to use terminology that has been around since 1984. Another common situation will be, and this falls into the category of incomplete biomarker information. Another common situation is that if imaging is used for characterization of research participants, then tau PET may be missing because it's relatively new and hasn't migrated throughout the research world yet. It will. It's on its way, but there's not obviously complete penetrance. And in these situations, if someone is amyloid positive, they simply are labeled in the Alzheimer's pathologic continuum, and if they're negative, they're not in the Alzheimer's pathologic continuum, without doing further designation of Alzheimer's disease, yes, no, based on tau. And finally, many research programs will only have access to MRI. So they will not have access to PET, they will not have access to lumbar puncture, or PET and lumbar puncture are not feasible for whatever reason in those programs. And we're not saying in any way that this kind of research is not valuable. MR has a lot to contribute about cerebral vascular disease and also providing prognostic information. So in conclusion, we hope that this research framework will assist with the research agenda. We believe that it will provide a better understanding of the sequence of events that lead to cognitive impairment, a better understanding of the multifactorial etiology of dementia, which has multiple causes, not just Alzheimer's disease, a more precise approach to therapeutic interventional trials, as Paul has indicated. And finally, we recognize that the validity of this framework needs to be assessed in more diverse populations because, by and large, biomarker studies to date have been done largely in people of of European ancestry, upper socioeconomic status, and not in population-based, but rather clinic-based cohorts. So the last thing I'll mention is that our work group is interested in feedback from the public. What do you like? What do you not like? What do you see as a stumbling block? What do you see as an advance in this proposed research framework? The research framework is not complete. It's still in evolution. It won't be completed until sometime later this year, probably around December or so. The working document, which I've alluded to earlier, is publicly posted at this website you can see in this slide here. You can download the document, read it over, and please send us any comments or suggestions, again, back to this same web address. That's my last slide, and thank you for your attention, and again, thanks for inviting me along with Paul.
Great. Thank you so much, Cliff, for that really comprehensive presentation. And I know that we were really pleased to see that the working group is open to receiving comments from the public, and so we appreciate you including that slide in your deck. We know we're a little bit over time, but there are actually two questions that came in, and so I'm going to jump right into those. The first question I'm going to direct to Paul first, and your line's open. Are cognitive biomarkers and tests proven to be accurate at teasing out Alzheimer's disease progression from normal aging in the earlier prodromal stages of disease, and how has this been investigated?
That's a great question. I think that cognitive measures at the very early stage are not specific in and of themselves. Again, we place people on the AD continuum using a biomarker, using amyloid measures, either amyloid PET or CSF. And once people are on the continuum, we find it very useful to measure their position using a cognitive test. But in the absence of an amyloid biomarker, no, cognitive tests by themselves are not specific enough at the early stage of disease. As we get towards the middle and later stages, there are characteristic memory impairments, amnestic impairments, that can make it likely that someone is on the Alzheimer's continuum. But even then, without a biomarker, and Cliff mentioned this, without a biomarker to indicate amyloid pathology, there are many errors, a high percentage of false positives and false negatives. So no, I do not think that cognitive measures alone are diagnostic.
Great. And I think that actually addresses another question we had as to whether or not the new definition would apply to other forms of dementia like FTD, where any of the biomarkers have not been present. There's one more question, and then I think we'll close. The second question is, and I'd ask either of you to answer this or both. The new way of looking at Alzheimer's as a continuum rather than a symptomatic condition is in some ways it's considered to have someone having the disease prior to the onset of symptoms. But beyond the value of this approach for research and the benefits it's going to have on drug development, is there anything that you think that patients and their families need to be prepared for, given that the presence of biomarkers alone and the absence of symptoms means that you technically have Alzheimer's disease?
If I can just say a couple of things about that. I don't think that everybody should go out and be tested to see whether they have amyloid at this point in time. I think, however, that things are going to change in the coming years, and I think that we are going to develop treatments that change the course of the disease, and when those treatments are available, I think it's going to be important for everybody to know whether or not they are on the Alzheimer's continuum. So at that point, it presents an opportunity to get early effective treatment. Today, these biomarkers are very useful in our clinical research, our observational research, and our drug studies. I don't think that this framework should be applied to the population now. I don't think that aging individuals should be seeking biomarker tests, not until we have intervention. So at this point, I don't think there's a significant benefit.
That's really
right.
helpful.
So this is Cliff, and I completely agree with what Paul said. There's no indication for this widespread testing of cognitively unimpaired individuals right now. But when treatments become available, then it will be not just a nice thing to do, but it will be essential, because the bottom line is, as Paul indicated, clinical symptomatology is not specific, and people should not be treated for a disease they don't have. And treating people, let's say with anti-amyloid interventions, without biomarker documentation that they're on the amyloid accumulation pathway is not a great idea. Imagine treating someone for cancer with chemotherapy and not really knowing whether or not they actually have that cancer. I mean, it's unheard of. And it would be equally unheard of to do that, I think, when treatments become available for Alzheimer's pathology.
Great. Okay. Well, we are well past time, and you've all been very generous with your time. So I'd just like to wrap up by thanking you all for your attention, and thank you, Cliff and Paul, for your very informative talks. I just want to call your attention again to ACT-AD's upcoming meeting on November 16th. It'll be our 10th annual meeting with the FDA. We already have leadership from the Division of Neurology Products and the Division of Psychiatry Products confirmed to participate in the meeting. And as I mentioned earlier, it's going to be very focused on the earliest behavioral and neuropsychiatric changes that occur in Alzheimer's disease. We expect that the draft agenda for the meeting is going to be up on ACT-AD's website in a few weeks. But if you'd like additional information in the interim, you can contact my colleague, Ryan Carney, and his information is up on the screen right now. We know that many of you who registered for this program are already registered to participate in the meeting, but if you haven't, we would love to see you there. And then again, I'd also like to thank ACT-AD sponsors for their generous support of the coalition and for making the webinar possible. And I hope you all have a great afternoon.